ಎಂಟರ್ ಸ್ಕೀಮ್ ತರ ಮಾಡ್ಬೇಕಾ? Okay. Ah, 10 years direct. Huh? 6 years. ನನ್ನ ವಾಯ್ಸ್ ಆಡಿಬಲ್ ಇದೆಯಾ ಮಂಜು?
Yes, sir. Yes, sir. It's audible. Sir, as told participants, we will start at 45. So we will wait for 4 minutes more.
Okay. We will wait.
Manjunatha sir, kindly start with the introduction.
Okay. Good afternoon to all. Okay, and I welcome Dr. Girish sir for this session. Girish sir is a principal scientist at our regional station in plant breeding. Sir did graduation and post-graduation from UAS Bangalore, and sir did PhD from ICR IRI New Delhi. And the major achievements include sir has developed several rice varieties, that is the TR-done 55, 56, 58, 60, 17, and sir has also associated in marker-assisted transfer of imagitape resistant herbicide gene to DR-done 60, and he has developed several rice varieties. And sir has published several around more than 60 research papers in the reputed journals having high NAS rating and impact factor. Sir, I welcome you for this session, sir. Kindly start your presentation, sir.
Thank you, Manju. Thank you very much. So very good afternoon to all of you. Hope I am audible.
Yes, sir. You are audible, sir.
Okay.
You are fully prepared.
I'll be
Please
presenting on
continue.
genetic analysis and molecular mapping of early seedling vigor on weed cooperative traits under direct seeded rice conditions. So I'll be going through what is the importance of direct seeded rice and early seedling vigor and weed competitive traits, and what are the traits that we need to target, and what are the breeding approach that we need to employ in order to develop the weed competitive trait varieties, and what are the work we have done at Indian Institute of Rice Research while I was working at that institute. I'll briefly going through and I'll presenting the methodologies and also the work and the results. Okay. So you know that rice is a very important rice crop, not only in India, in the entire world. In India, more than 47-48 million hectares is cultivated, and there are two cultivated species, Varieja sativa and Varieja glabruma. Both are cultivated species. Varieja glabruma is an African cultivated, while the Varieja sativa is the Asian cultivated species. So there are two species. In addition to that, there are also 22 wild species of rice are available. So rice is a very important crop. So as I said, we are producing more than 135 million tons of food grain production. So we are not only food sufficient in food grain production in India, but also we are exporting rice to the other countries, including the basmati rice. So rice is a very, very important, not only in terms of food security and also nutritional security. Okay. So with this, so the main rice, mainly it is cultivated in transplanted rice cultivation. You all know that the rice is a mainly water consuming crop. It requires a huge water in order to cultivate through transplanted and irrigated methodology. So, but as your days passes, now there are global warming and climate change. So water is becoming a very important element. Day by day, water availability for agriculture, particularly for rice, it is reducing. So in order to address this water shortage for rice cultivation, it is essential that we need to implement or move towards the direct seeding rice. Where in direct seeding rice, we will not go for the transplanting and nursery preparation followed by transplanting will not be carried out. The seeds will be directly sown into the field conditions and crop will be raised, and rest all the package of practice, management practice, rest all by and large is similar to the transplanted except nursery preparation and transplanting. This has will result in saving of lot of labor which required for nursery preparation followed by puddling and transplanting, and also huge amount of water which is required to maintain the water stagnation condition for preparation of puddling and also nursery preparation, and also during the throughout the crop stages we need to stagnate water under transplanted condition. So these two major advantage. So these are the two major advantage. Not only these two also. So when you compare to transplanted condition, in case of direct seeded condition, we can also go for mechanization because transplanted condition, mechanization, transplanting to some extent we can do followed by weeding, manning. But in case of direct seeded condition, right from the land preparation, even direct seeded drill based sowing cultivation we can take up. So and also weeding. So there is a huge scope for mechanization. So there is opportunity where the many mechanized company can participate in the rice cultivation practice. So with all this advantage, so direct seeded rice is gaining its importance, particularly in the post-COVID era. During the COVID, you all may be knowing that so there was lockdown, lot of use and cries in terms of labor ability, agriculture. So people are forced to go for direct seed condition, some particularly Punjab, Haryana, and some parts of southern India also, where people are forced to go for direct seeded, despite the fact that they were not much into direct seeded condition, situation has forced. So that has awakened not only the farmers but also many policymakers that direct seeded condition has a huge advantage as compared to transplanting, and there is no much yield penalty. Rather, no yield penalty. There is no much. So most of the varieties which are being irrigated cultivated now they are mainly there. They can be cultivated in transplanted condition. Although there are no varieties which is specifically bred for direct seeded conditions. Okay, so there is a need that so the variety always have a adoption limitations. The variety which is bred for a particular ecology always perform well under that particular ecology. For example, if you develop a variety for transplanted condition, they do perform well under transplanted. But under direct seed condition, they may not do the extent, they may not able to express full potential, genetic potential. Therefore, it is essential that we need to adopt a variety which is specifically suitable for the direct seed condition. There are a couple of major disadvantages. The major disadvantage rather than the major challenge in direct seeding condition is one is the early seedling vigor. Most of the irrigated ecology bred varieties, they have a poor early seedling vigor. So their adaptability to the direct seeding condition so is a means imbalance. So there is a need to develop a variety which has a very good early seedling vigor. So that is one requirement. Another one is in direct seeding condition, weeds are a major problem because in transplanted condition we are going by puddling for stagnation of water. So weed may not grow to the extent, but in case of direct seeding because of aerobic situation, the weed grows much faster and weed population density will be very high. So these two are the major bottlenecks for the development. I mean, using large scale adoption of direct seeded condition. So whereas the glabrima, as I said earlier, it is one of the cultivated species. Only in Western Africa, it is largely cultivated. The origin from Varaja Bhartai. So it has the inherent potentiality for early seedling vigor, and also they thrive well under the undernourished conditions, and they have the weed competitive ability. So there are many advantages. So these are the trait actually which is essentially required for developing variety under the direct seeding condition. So we thought that this should be utilized as a donor species. That means that there are whatever the traits which are like weed competitive, early seedling, which thrive well under the undernourished conditions. So like nitrogen fixation, these are the some of the traits and rapid canopy establishment, one of the weed competitive traits. So rapid establishment, high seedling biomass, higher root growth. So these are trait we wanted to target. So what we did actually, so this was the user as a donor parent. So what we made is we attempted a cross between IR60 per, which is very popular variety in the irrigated ecology, medium duration variety. So IR64 was made a cross with Varese glabra. So Varese glabruma is will be a donor, and this is Varese sativa. These are the intergeneric crosses, so interspecific crosses. Okay, so these interspecific crosses were attempted, and you know that F1 was fully sterile because of the compatibility issue, because of sterility genes. So F1 was fully sterile. So we attempted backcross populations. We went up to 3 backcrosses followed by selfing, and we developed the BC1 F2 population. So BC1 F8 population or F9 population we developed through backcrossing, the repeated backcrossing followed by selfing. So this we did, and then the backcross population, what we did, we phenotyped for weed competitive ability, early seedling vigor at 15 days after direct seeded sowing, and also 30 days after direct seed, and also we attempted yield attributing traits. So these are the some of the traits we did. And also what we, in addition to what we did actually, we also went for genotyping of population. We used the 2 methods of genotyping. One is 1 which is having 1000 SNP marker loci. So in a population, entire population or genotyped with 1 In addition to that, in order to increase the density of markers, we also went for a genotypic by sequencing approach. So where the entire genome was sequenced using GBS methodology, and then both the SNP markers, both the marker system, genotyping platform like 1 and GBS, both were combined, and you have used that to develop the linkage map or linkage map, that is integrated linkage map. So with both phenotypic data on the one side, genotypic data on the other side, both were used, and we are subjected to QTL mapping, QTL analysis. Then we identified the candidate genes. This I will be presenting in the coming slide. So then we also identified some of the major QTLs, which is very important, which we identified for the our derived from Varese glabruma. So this I will be presenting. So as I said, as I explained, so phenotyping, so we mainly targeted seedling vigor traits, yield attributing traits, weed suppressive ability, weed tolerance, and also some of the other important traits. So we used BC1, F1, F8, F7, F8, and F9 population, 3 generation. Once the population reached F8, so these are all mostly fixed generation. We had around 184 lines. So generally, we employed augmented design, and the phenotyping was carried out under 3 conditions: weed-free, weedy, and partially weed conditions. So all the conditions, we also used some of the checks like Samosuri, Sabita, Sabagidan, very popular variety, Brown Gara, which is landrace, but having a very good tolerance to weed, and Drathan 44, which is a very popular variety, cultivated for particular direct seeding. It is gaining a lot of importance. And other some of the parental lines like IS64 and CG235 is the Varese glabruma. So this we did for 3 seasons. So 3 seasons we phenotyped, and these are some of the traits as I explained already. Some of the parameters which I have used for phenotyping, like early seedling vigor, like germination percent, number of leaves, etc., etc. So these are some of the parameters we estimated. The whole population around 192 lines were almost phenotyped for all these traits under 3 conditions: weedy, weed-free, and partial weed conditions. And also, in addition to yield, we also went for weed suppressive ability and weed tolerance. Weed suppressive ability means it is the ability of the plant to suppress the ability of the rice plant to suppress the weed growth. So that is based on the biomass of weed we will take, and then we will try to estimate the weed suppressibility. Weed tolerance is it is a economic threshold level of yield loss. How much yield loss actually we are not losing, how much yield we are not losing despite the presence of weed. So that is the weed tolerance. If weed is there and we are not losing any yield, economic threshold level, maybe that we can say that this is not having any, this is having weed tolerance. These are some of the parameters we phenotyped. These are some of the glimpses of the field photographs which are taken in the direct seeded condition with the weed, without weed. So like, I'm just presenting some of the graphs, like how the phenotypic values were. ESP means early seedling vigor at 15 days under weedy and weedy free conditions. These are some of the parameters which are showing normal distributions, and we can see there is a significant difference between the weed free and weedy conditions for these parameters. Again, for the 30 days, ESP1 and ESP, early seedling vigor 1 and early seedling vigor 2. These are the very two important parameters which are routinely used to estimate, to understand the seedling vigor parameters. So these are the, you can see the difference and also see the normal distribution. These are the some of the photographs. So this is again, this is another season. What I explained, the first one is the 21 Rabi and this is 22 Kharif. So these are the some like grammatic representation, graphical representation of our phenotypic data. This is the first one is 15 days. And again, this is another 30 days. You can see the normal distribution of curve and the various other parameters which are represented. So we can see the difference in terms of seedling vigor and also other parameters. This is, so this is 2023. Next is the third season, 2022-23, under weed-free weed conditions. Again for 15 days and 30 days, seedling vigor 1. I'm only presenting two parameters, seedling 1, seedling vigor 2. But also you have taken so many other parameters, which I am not presenting because it is, I mean, it will take a lot of time. So these are some of the photographs, like we can see the rice plants, and also we can see a kind of a wheat truss. We have see on the right side corner end, you can see the glabrum line, I mean, doing good. I mean, surviving even despite having a huge this. So for information, you know, when weed is there, actually rice won't survive. When you complete weed, the weed is, if you don't do weeding, what will happen? At certain stage, plant completely killed. So because of toxicity, kind of phytotoxicity chemical which is released by the weed, actually kills the entire plant. But surprisingly, particularly this glabrium accession, which you are seeing CG225 on the right side down on the last plant, where you can see this particular plant able to survive. So that's how it has a weed tolerance. That's why we use this particular parent. Why we wanted to go, what exactly, I mean, what exactly it is there, whether it's genetic potential or something else. So this is just a glimpse of some of the seedling parameters. Photographs we have taken, like you can see the root length and shoot length and leaf biomass, other parameters you can see in the photographs. So these are taken up to 30 days. These are, first one is at 15 days, some are at 30 days. Now this is at 30 days. These are some of the introgression lines, K34, 832. These are the lines which are from, I mean, the real lines, so which are showing a good performance at different stages. You can see like parents and also Sabita. These are the checks actually. BPT 5204. BPT 5204 is irrigated ecology. So under direct seeded condition, it won't do good, at least in the early stages. So these are some of the, again, photographs showing, I mean, different conditions. So frequency distribution curve, again, under weed-free conditions, Kharif 2022. These are the like other parameters. Earlier I explained about early seedling vigor and other things. Now we are yield parameters, like single plant yield and test weight. These are the first one is 2022 and 22-23 season, different seasons. So these are the some of the traits we have estimated. Now these are the descriptive statistics and weed tolerance and weed suppressibility. You can see overall 55, 30%, the difference which you have noticed for weed tolerance. So distribution of weed suppressibility trait under different conditions. You can see level of resistance that we noticed in the population as compared to the recurrent parent IR64, 75% resistance, weed suppressive ability, and that is probably because of the huge biomass. So these are some of the graphs distribution of weed tolerant trait during two seasons, that is 22-20 and 22-23. These are some of the photographs. So these are all about what so far I explained about is phenotyping. Now with that phenotypic data which you had a population, so we wanted to go for QTL analysis, identification of QTL associated with those traits using those phenotypic data. So as I earlier explained, so we had the genotypic data. We genotype the population with two genotypic platform. One is one Kerika, another one is genotypic by sequencing. So with both the popular genotypic platform combined together, and this is what is. So totally we used around 2000. I mean, we used, these are the number of markers in one Kerika, around 787 marker were there. And in case of GBS, we got this is after doing all the statistical analysis, like whatever deletions and other things, we removing all the unwanted loci with the frequency, rare allele, all we did. So we used around 2200, like 2087 9 marker ultimately, which are actually found polymorphic between the both appearance. These are the final list of marker which are used for our linkage map construction. So 2089 were marker. So this was actually used with a linkage distance of 3000, 33579 and the total distance. So we used this for linkage analysis. So this is the linkage map. So this is the linkage map. So which are distributed across all the chromosomes. So rice has 12 chromosomes, so 12 linkage groups. So these are this. So with this we went for QTL analysis. So ICIS mapping and QTL color profile, these are the standard protocol which we use. So we got a large number of, both major and major SN, because we used more than 35, 40 tries. So lot of QTLs we got. So we with this, the idea was to how to go ahead with these markers. We wanted to reduce the number of QTLs which are in terms of major QTL, like more than 10% R square value. We wanted to shortlist, and we wanted to identify the QTL which are actually common across the trait. So we cannot use this many QTL for any purpose. It's of no use unless we do further analysis. So what we did actually, we try to identify the QTL which are actually common across the seasons, and there are QTL which are actually working across the different traits. That was our idea. So these are the number of QTLs, and we wanted to remove all the minor QTLs. We wanted to only concentrate on the major QTLs, which is having R square value more than 10, and also the season wise, if you take common across the season under weed, and also not only season, we wanted to go for under weedy and weed free condition which are common. So that how we narrow down the number of markers which you got here. So huge number of QTLs which you got. So we narrow down to almost 12 marker we got to. So 12, 15, so common markers, that's what we want. So with this, so these are some of the parameters I just was explaining. So what we did, that is why again it is the same repetition. So we had around 248 markers, 2 QTLs we got, which are related to 20 for late seedling vigor, which is late seedling vigor means actually seedling vigor we have taken at 3 stages. So one is at 15 days and another 30 days. Late seedling vigor we wanted to do it at 60 days because prior to the flowering, which is very important at the 60 days. So this is what we wanted to do, because biomass accumulation at 60 days is very, very important. That is prior to the flowering stage. So these are some of the QTL we got, and ultimately 39 major QTL we had from entire thing. So 9 had a positive effect. Again, how many of them actually contributed from glabruma? So that was we wanted to have. So like this we narrow down the QTL. So this is again the same as I explained under VD free conditions and VD condition. These are some of the information I have provided. So then we wanted to, like, once you narrow down the major QTLs, and then we narrow down the QTL which are common across the season and also VD and weed free condition. So we got around, among them again we wanted to go for QTL hotspot. QTL hotspot means co-localizer QTLs, like the QTL which are located in the same region but controlling many traits. So there are QTL hotspots or co-localized, like we got around 18 plus 6, so 24. So again we have to narrow down. So what we did actually, from among them 24, from again particularly 18, so we narrowed down to 6 QTL regions. So we considered under VD and weed free conditions. This QTL hotspot actually common across both VD and weed free conditions, and they're affecting nearly 12 traits. So these are the 6 QTL we narrow down. So these are the 6 QTL which QTL region rather we should say, because these are the regions they're affecting multiple traits. So these are some of the parameters like R square value, additive variance, and other parameters which are associated. This is just table descripting about. So we wanted to go further. So what we did, so these are the just representing in terms of chromosome positions. So those QTL 6 out of 6, 5 it is represented QTL positions on the particular chromosomes, like chromosome number 2, chromosome number 4. So like this it is represented. So 12, 8, and 1, 4, and 2. These are some. So then again, what we did actually, we wanted to identify what are the candidate genes. This is in silico analysis. So based on the sequence information which is available, based on the SNP positions, we went for in silico analysis in order to identify what are the candidate genes actually they're present in that QTL region. I mean, in the marker region, exactly. Okay, in the marker region. So these are the some of the list I am presenting. These are the actually present. These are the some of the regions. I mean, among the 7 regions. So these are the different QTLs which are actually present. So, so these are the some of the candidates we identified. So major thing is in this. So this one is genome recovery percentage. So we need to have based on this QTL whatever you have got. Actually, we can estimate because we have the information for SNP genotype. So we have the entire genome recovery percentage, and we need to be evaluated for stability of this QTL, because whatever QTL which you got that need to be evaluated for stability, and also validation file mapping, because whatever 7 regions which you have got, so we need to also validate in alternate population. We need to design kind of a cache marker for this. This work need to be carried forward. We need to design cache marker for this, and particularly the major QTL detect for WA center need to be further validated. So these are some of the findings from my study. So this is all about my presentation. Thank you. Manju?
Sir, thank you, sir. Thank you,
think
sir, for very nice presentation, sir.
Thank you.
I request participants for any questions and clarifications. Any questions from participants?
Yeah, please.